right, so today we will talk about clustering as an exploratory technique for visualizing data, like our data, multidimensional, high-dimensional omics data. Okay, so I posted a couple of new resources on eCampus. So under Notes, there is now this PCA and clustering, and there is this Introduction to Statistical Learning. So PCA and clustering is what I'm about to open right now. Show you what it looks like. The file itself is called unsupervised.pdf. That's because the slides are presented as unsupervised learning. And I'll comment on where that comes from in just a second. But these slides were written by the authors of the second resource here. So second resource is Introduction to Statistical Learning. And this there's a website. Gareth James is one of the authors, and on his website you can get the PDF for the whole book. Tipshirani and Hastie are famous statisticians at Stanford, and they've written two books. One of their books was called The Elements of Statistical Learning, which is kind of a classic in machine learning / statistical learning. And then this book is the newer version that is more user-friendly. It's easier, more accessible, and it's got lots of R in it. By the way, Daniela Witten is one of the co-authors. She's now, I think, an associate professor at the University of Washington, Biostatistics Department, and she will be here in a week or two. She's going to give a talk. She was given an award she won an award that was given by Professor Raymond Carroll in our department, Young Investigator Award. So if you're interested in this stuff that you're going to see, you might come check out her seminar. It is. Yes, you can download it. And it is right. Well, actually, this is the whole PDF right here that I put on eCampus. And if you open, I'll download this too. If you open the book, here's the book. Statistical learning, this learning idea, shares a lot with machine learning. You know, depending on who's doing the talking, whether it's a computer scientist or a statistician or an engineer or something, we might use different terms to mean similar things. But the common denominator is that we are taking data, and we are either doing predictive modeling, like I want to take omics data, thousands of variables, and I want to use those to predict whether somebody has cancer or not, for example. That's predictive modeling. That's supervised learning classification. Supervised in the sense that we have a known quantity. There's a right answer that is known for the data we have. Here's 50 people for whom I have sequence data, and for each of the people, I know whether they have cancer or not. So training data is what we need in order to do supervised learning. When we don't have training data, like we don't know the class membership, cancer versus not cancer, then it's referred to as unsupervised. And principal component analysis and clustering are examples of unsupervised learning. Because when we did PCA, we didn't say, let's break it into these defined groups. We were more looking for what are the groups. When we do clustering, same thing. We don't know what the groups are. We're looking for structure in the data. We're trying to figure out, are there any groups? So the relevant part for what we're going to talk about right now is down in Chapter 10, I think. Chapter 10 is all about unsupervised learning. Most of the book is about predictive modeling. So it talks about linear discriminant analysis and other simple type methods through to more complicated methods like support vector machines and random forests and stuff. But they all are doing basically the same thing. Give me input variables, we will predict class membership. So check out the chapter on unsupervised learning if you want to read ahead and get some more details on either or, PCA or clustering. We're going to talk about two flavors of clustering, k-means and hierarchical. And I'm going to start with hierarchical clustering. So I'm coming back to the PowerPoint slides here, and I'm going to go down. The first half or more of the slides are about principal component analysis, so you could read through that if you wanted to revisit what we've talked about in class. And let's come to clustering. I'm going to go down to where we start talking about hierarchical clustering. So here's the idea of hierarchical clustering. Suppose we have 5 observations in 2 dimensions. That means we have a sample of size 5, and for each of our individuals, individual A, B, C, D, and E, we have 2 variables that we're measuring. So it might be height and weight, for example. I have multivariate data because I have 2 variables that are being measured for every individual. If I just had one variable, if this was univariate, then in terms of asking about clustering, what clustering is about, I could just make, let's say, a dot plot. Just put a dot wherever there are observations. Make a picture like that, and I can kind of see, quote, clusters, clumping of data together. One dimension, you can just make a picture like that and look at it. 2 dimensions, it's not that much more difficult because we can just plot it in a scatter plot. So in 2 dimensions, like in that slide that we were just at, we could draw individual 1, 2, 3, 4, 5. So here's variable 1 and variable 2, height versus weight or something. We could look at this picture and look for, quote, clusters. But what happens when we get to 3, 4, 50,000 dimensions, like with the cardio or the whatever the data is in our homework? We have 55,000 features. So if I wanted to cluster the individuals now, I can't make an easy picture like this. So I need an algorithm that does something sensible. What are we doing when we do this clustering thing? Intuitively, you know, when we started over here in the one-dimensional case, we were just looking and asking, are there locations where there are points that seem to be more similar to each other than to other points? An alternative picture could have been, you know, like this. in which case there's not really any distinct locations. Everybody's just kind of in the same group. I don't see clusters. I don't see evidence of clusters. Okay, so in general, suppose we have P variables. P is equal to 2, 3, ... 55,000. What I want to do is ask about find groupings of individuals that are similar to each other, more similar to each other than they are to other individuals that aren't in their little cluster. So in order to do that, I have to get specific about what I mean by similar. So individual A, how similar, how close is individual A to individual B? Is individual A closer to individual B, for example, than to individual E? In this case, yes, and you can see it. But to be general, what do we mean by closer? Well, we're talking about distance. What's the distance between A and B? And what's the distance between A and E? So if I have multivariate data, I can define distance in a multivariate way, like as a vector distance. What is the distance between these 2 points in two-dimensional space? So if we have, like, if the coordinates for A are V1, let's say V1 for A and V2 for A, those are the height and weights for individual A. And the height and weight for individual B are V1 for B and V2 for B. Then the distance, what is the distance between point A and B? We could define that in a variety of ways. One simple way to define distance is the usual vector definition of distance, or the Euclidean distance, which is the square root of the component differences. So, like, what's the difference on variable 1? Square it, and add that to the difference on variable 2. Squared. That's Euclidean distance. But there are other ways to define distance. There's a whole listing of ways to define distance. So step 1 when we do clustering, I mean, the main thing is that we need to be able to talk about how similar, how close are these things to each other. So we need to define a distance. So let's come over to the slides. Let's go through this real quick. The idea is in hierarchical clustering, it's just like what I was drawing on the whiteboard. We have 5 individuals and we have 2 variables. Who's closest to who? Step 1 in the algorithm, hierarchical clustering is an algorithm. It just goes through and follows some basic computing rules. Step 1 is to find the 2 individuals that are closest to each other. Out of all the individuals, do all pairwise comparisons. Look at A to C, A to B, B to D, E to A, all those. Which pair is closest? And in this case, it's A and C. You can just see it from looking at the picture. So step 1 is A gets joined to C, and we now have a cluster. Our first cluster is A and C. Everybody else is by themselves still. Step 2 is to repeat. But now A and C are considered as together. There's one fewer individual, so to speak, in the data. So now we could ask who's closest. And now it was D to E. One complication that just arose as we went from this slide to this slide. When we came to this slide, we wanted to again ask who's closest. So I could ask, how close is D to E? How close is D to B? How close is E to B? What about A and C? We are treating them as a single entity at this point. They are a cluster. I still need to ask about their distance, the distance from this new entity to everybody else. So for example, I'd like to know what's the distance between this cluster and individual B. We've already talked about distance. We have to have some definition of distance. One possibility is Euclidean. Here's the second issue or detail that we have to handle when we do hierarchical clustering. How do you define the distance between this cluster that has two things in it and something else? That is referred to as the choice of linkage. What linkage to use, and I'll talk about that in 1 second. So just to go backwards for a second, we have these 5 individuals. Step 1, who's closest? A and C are. So step 1 is to join them together. Next, reevaluate. Now who's closest? A and C are one thing now, though. In this case, D and E are next closest, so they get put together. From there, do it again, and now the closest objects are individual B and the cluster containing A and C. So those 3 things get joined together. And then there's only 2 things left at that point, and there's only one thing left to do, which is join everybody. So with hierarchical clustering, we get a picture like this one. This is what we'll end up looking at. It's called a dendrogram, dendrogram. And let's talk through what it's showing us. The fact that A and C, usually we won't have all the coloring and the dashed line segments and stuff. I'll show you one in R in a second. Usually we just have like a tree-looking thing without the coloring. The lowest merge point was between A and C. So A and C were merged first. The fact that their joint is closest to the bottom tells me they were closest out of everybody. These are the most similar 2 features. The next closest features were D and E. They were joined with a merge point that was a little bit higher than A and C. After that, B got lumped in with A and C, and after that, everybody got piled together. These heights, like the numbers on the vertical axis, are distances. Whatever we define distance to be, like Euclidean distance, the fact that this join point is located vertically at about 1.1 says that the distance between individual A and C is 1.1. And the fact that the B joining with A and C is up around 2 says the distance between B and the cluster containing A and C is 2. Okay, so let me tell you a little bit more about linkage, and then let's go do a real example and interpret it. The idea is you take data, you don't know about structure, you just have omics data. Like the yeast data, we have thousands of genes, and then we have 20 individuals or 20 arrays. And step 1 with data is just to look at it and explore it. Univariate data, you might make histograms, box plots, stuff like that. Multivariate data, it's harder to pick it up and look at it. So it's handy to have some summarization tools or visualization tools like PCA, like clustering, because now you can take a big old matrix and boil it down to a picture like one of these that is informative. So we're going to make one of these and see if we can learn anything based on the picture. So before we go actually do this, there's 2 issues that we have to address if we're going to do hierarchical clustering. First is, how do you want to define distance? Let's see if they have a slide about that. So here's a slide that talks about distance. Default choice is Euclidean distance. That's what will happen in R unless you tell it to do otherwise. Right. I'll show you how to compute it in a second. That's default. There's something called the Manhattan distance. There's something called city block distance, which I think is the same. Canberra distance. There's a bunch of different distances. I don't know why I would recommend one of those over the other in most cases. Every time I've done clustering, I either do just the default Euclidean distance clustering or a correlation-based distance. Okay, so let me talk about differences here. Euclidean distance, it takes the actual raw values of your data and just takes differences and squares them. That's the basis for the distance. So suppose you have, let's see. Suppose we have two individuals and well, let me look at this picture here. Suppose we have three individuals. Consider three individuals, and for each individual we have 20 genes on one of these samples. So I have a 20 by 3 matrix of, quote, omics data. How close is individual 2, or let's say individual 1 to individual 3? For each of the individuals, I have multivariate data. So close requires that I compute a distance, a vector distance. If I were to do Euclidean distance of these two at the bottom, they would be pretty close because all of their individual points are pretty close. So like the distance, the Euclidean distance between individual 1 and 3 is pretty small. If I were to do Euclidean distance between, say, observation 3, which is the pink one, and observation 2, which is the green one, because the magnitude differences are big, like from here 15 down to about 2, that's a big number, square it, gets even bigger. So the Euclidean distance between the green and the pink would be big. But do we want that to be the case? Here's three individuals. We're studying something. Suppose these are genetic, well, let's not say that. Let's just say we have three individuals and we want to group them somehow in terms of similarity. Okay, individual 2 and individual 3, their absolute numbers are different from each other, clearly. But what about the pattern? The pattern for individuals 2 and 3 looks similar. If you follow from left to right, there's this cyclical peaking and this pattern. There's a pattern, and it looks pretty similar for both of the individuals. So one option would be to say, I don't care so much about absolute differences. Like think about genetic expression, the expression level of genes. You have some genes that are much more active than other genes. They're pumping out more copies of their protein than other genes just because they are needed in higher quantities. But maybe they follow similar pathways and respond similarly to stimuli. Cancer hits, and here's some genes that all do basically the same thing. But some of them are more active than others. So the absolute levels, the absolute numbers that you see for these different genes might be very different. So their distance, their Euclidean distance might be very different, but their correlation might be close, high. Because if the pattern is similar, correlation doesn't care about scale, it only cares about correlation. So one way to do clustering that has some nice things going for it is to use correlation as your distance. How close are two individuals? I could define that as what's the Euclidean distance. Or I could define it in terms of how correlated are they. So for example, if I'm looking for structure in my data, maybe I'm looking at time course data. I have, over the course of the life cycle of a cell, I have measurements every so many minutes or something of expression levels, gene activity levels. And I'm looking for genes that do similar things over the course of the cell cycle. Young cell, we need certain kinds of proteins, and then as it gets older, we don't need those anymore. So I'm looking for genes that do similar things. I'm looking for similar patterns. I would want a correlation-based distance. Quantify distance in terms of similarity of pattern, not absolute difference. So step one is to figure out distance. What distance measure are we going to use? Step two is, yes, go ahead. Yeah. Yeah. Okay, good. Question was, what about correlation between variables? What about correlation between variables? Let's see. There is a way to compute a distance and account for correlations. So looking back at what we did on this screen there, suppose for individual 1, we have y1. So that might be 50,000 numbers. Intensities or gene expression numbers for 50,000 genes that are on my array, say. For individual 1, here's the one for individual 2. And what's the distance between these two individuals? If I were going to do Euclidean distance, then I could write the distance between y1 and y2 as y1 minus y2 transpose. So y1 and y2 are vectors right now. And this is vector arithmetic. Take a difference and kind of multiply it by itself again to square it. And remember that if I have two vectors u and v, if I do u transpose v, that's the sum of their component-wise products. That's an inner product. So this expression is the same. It is a way of saying Euclidean distance. This expression can be modified to weight and incorporate variances and covariances if I put something in the middle. So if I do y1 minus y2 transpose, and suppose we can estimate a variance-covariance matrix, which we can do, a covariance matrix like var of the data set object in R, gives me a p by p covariance matrix. So variance is down the diagonal, covariance is on the off diagonals. If you do something like this, this could be referred to as a statistical distance because we are allowing for differences in variation and we're allowing for correlation. So you can imagine, here's a 2D scatter plot of data. Or let's just imagine that's where data lives for a particular case. Variable 1, variable 2. So when you sample from the population, you're seeing points that tend to be in here. What about, here's the center of the distribution. And what about like this point right here? How close to the origin is that point? In Euclidean sense, it's like that. Here's another point that is the exact same Euclidean distance from the origin. If I did Euclidean distance to the origin with red and green, they'd be the same. But in terms of this distribution, correlations and differences and variances, which of red and green would you say is closer to the origin? In terms of which of red and green would be more likely, less unusual? In a statistical sense, is green or red closer to the middle of the distribution, do you think? Green, because it's in the cluster more. Red is on the outskirts. Even though Euclidean distance is the same, in a statistical way, you wouldn't really call them equally different. So by doing this, the distance for the red guy would be bigger than the distance for the green guy. So correlation and stuff is an important consideration. You can accommodate it by using a statistical distance. You might see this referred to by a pretty awesome name, Mahala Novus distance. Tell your kids about that. They'll be excited. Distance is important. The other piece that we need to talk about briefly before we go actually do this is up here. It's easy to say what's the distance between individual A and individual B, and distance between A and C. But once we put, say, A and C together, the next step is to ask about distances again. So E to D and B to D, those are easy to do. But now I have this new entity that is composed of two things, A and C. And for example, what's the distance between B and that new entity, A and C? That has to do with linkage. How do you define distance between clusters? Here are the main, we're just going to focus on the first three. These are the main choices for how to do this. They're called complete linkage, single linkage, and average linkage. And if we look back at this case, let me just redraw the relevant piece here. Let's suppose we have A and C, and then we have let's do B. Step one in the algorithm is to join A and C because they are closest. Step two needs to consider B to this cluster. What's the distance between the cluster containing A and C and individual B? What we can do without problem is compute the distance between A and B and the distance between C and B. So I could look at the distance between, if in general we had, okay, so I could look at the distance involving the individuals. In this case, there's two individual distances. And depending on the linkage, I will turn this pair of differences into one number. In particular, complete linkage would take the maximum of these two distances. Single linkage would take the minimum, and average linkage would take the average. So if you want to know the distance between two clusters, like suppose now that it's A, C, and D, and over here is B and E. What's the distance between cluster 1 and cluster 2? I would need the distance between D and E, C and E, A and E. D and B, C and B, A and B. Get all of those, and then which of these linkage rules am I going to use? If it's complete linkage, you take the biggest distance between all those pairs. If it's single, you take the smallest. So which of those three would make it hardest to join two clusters together, do you think? Yes. Max, complete linkage, because distance according to that linkage is you're only as close as your two furthest components. So if there's two clusters and they're all really similar, except there's a couple that are very different from the two clusters, then overall those two clusters are very different. Single linkage is the opposite extreme. It makes it so that if there's at least one pair that is close between two clusters, you're going to call the clusters close overall. And average is in between those two extremes. There is no magic right answer for which one is best. They can give you different pictures and different results, unfortunately. Often I would recommend doing all three and looking at the results, and hopefully qualitatively the interpretation doesn't change too much. If the pictures look wildly different depending on what linkage choice you use, I would tend to not trust the pictures all that much because I don't know which one is the right one to look at. The ideal case is it doesn't change all that much. You get roughly the same story, and you don't feel so bad about having to pick one of these kind of arbitrary choices. So before we run out of time, let's go do this. Let's go to, say, the yeast data. And see where we are here. So I'm just running the code that we've run before, reading in the data, doing a little processing, and pulling out the median channel differences. Okay, and then down here is where we have clustering, and I'll just redo this for us here. The main function for hierarchical clustering in R is called hclust, as in hierarchical clustering. So I would do $hclust to start getting comfortable with it. And if you do that, we'll do that in 1 second here. So $hclust, hierarchical clustering, and there's one argument that you have to pass in. And that argument is, it's called a dissimilarity structure as produced by dist. What does that mean? We have data that we've loaded in. Capital Y is the main object right now. 6,740 genes or features, 20 individuals. So that's my data matrix. I'm not going to pass that to hclust. All hclust wants is the distances. It wants a matrix of distances, pairwise distances. So we can do PCA or clustering to look for structure among the individuals or among the variables or the features. It's a lot easier to do it with the individuals because there's only 20 of them. So let's do that first. So multivariate methods in R, if you pass in a matrix, it's going to treat it as if the columns are the things you're interested in. That's your variables. So if you do PCA on capital Y, you're going to get you're going to look at structure among the individuals. We're about to do clustering, and I want to cluster on the individuals. So what gets passed in, where did my help file go? What gets passed in is a dissimilarity structure as produced by dist, a distance matrix. So if we have, like back here, we have 5 individuals, we could define the matrix containing all distances as a 5 by 5 matrix. So let's put zeros down the diagonal because each individual is 0 distance from itself. But then on the off diagonals, like row 1, column 2, is the distance between individual 1 and 2, and so on. You can get that very easily using the dist function in R. So I'm going to compute distances. Distance matrix. I'm going to start by doing it this way. Dist is the magic function. So dist computes a distance matrix. This says that it is going to compute distance between the rows of the data matrix. So this is actually not following the convention that I was mentioning a moment ago. I want to look for structure among the columns right now. So I want a 20 by 20 distance matrix that shows all the distances between the individuals. As it is, I'm going to get a 6,740 by 6,740 distance matrix, unless I make the columns be the rows. So that's why here I'm going to transpose Y. When I do this, we're going to get a 20 by 20, actually not, T is transpose. It is not. T is just transpose. So we did dist. If you print dist, it has a special output. This is because a distance matrix is symmetric, and R is being efficient in the way that it's storing its information. Since it's symmetric, and since the diagonal is defined to be 0, really all I need to know and store is one of the upper or lower triangular part of this thing. So does that make sense? Hopefully so. Capital D is 20 by 20. So we have zeros down the diagonals because individual 1 is 0 distance from itself. Then we have D between individual 1 and individual 2, dot, 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 D1, 20, D19, 20. And the distance between individual 1 and 2 is the same as the distance between individual 2 and 1. And the distance between individual 20 and individual 1 is the same as that too. So this is symmetric, which means all we really need is one of these pieces. In order to have all the information, all we need is the lower part. That's what dist gives us. And that's all that hclust needs. Okay, I'll leave it at that. So we have a distance matrix, and then we just need to pass that into hclust. So I'm going to store the output because it gives me back some things that I want. hclust, here's your distance matrix. What else do you want to know from me? The other main argument is called method, and this is the linkage method. So we already defined distance. When we did dist, we chose the distance metric. We used Euclidean distance. That's what dist does by default. Now we need to decide on linkage method. By default, it's going to be complete linkage. So think of complete linkage as kind of like the most conservative way to do clustering because it makes it hardest to bring things together. Yes? It does. And I would know that dist gives me Euclidean by default by looking at the help file. So dist, there is a method. You can choose what method, and by default, because it says method equal Euclidean, that means that's the default choice. Down here, here's a list of the different distance metrics that are available. If you look at Wikipedia for hierarchical clustering, it'll tell you what these different metrics are. If you want to do correlation as your distance, you have to do it a little bit customized in R. I can show you how. There's also an example of doing that in this help file. So if you want to use correlation as distance, you have to be a little bit customized, but you can just use the example in the R help file. This gives you correlation. Like when we looked, where were we? When we were talking about distance, here, we said, how close are these three individuals at the bottom? If I used Euclidean distance, then the green one is very different from the others. But if I used correlation, because the pattern of the green one is similar to the other ones, correlation, if I quantify distance as correlation, correlation doesn't care about scale. It only cares about pattern. So if you want to highlight patterns and call things similar based on patterns, then you want to use correlation as your distance. If you want to do that statistical distance thing, don't know that there's actually a built-in choice for doing that. I didn't see it in here. You might have to get a little custom with that one too. And if you want to, I can help. Most of the time people do either Euclidean or correlation, in my experience. There might be other specialized areas where people do different things, but we are going to do hclust, and let's go ahead and we'll just go ahead and spell it out. We want to do complete linkage, say. When we do this, HC is a special object. It did come back with some items that we might be interested in. We can make a picture. That's the main thing you'll end up looking at when you do hierarchical clustering. Looks like a tree. It's called a dendrogram. And you can interpret it as what you're going to want to do is look at this and say, where are the clusters? The dendrogram joins everybody. At the top, everybody has been joined into one big, quote, cluster. But what you want to do is look at, for example, this grouping is separated a lot vertically from this grouping. These two individuals are not separated by a lot vertically from these two individuals. So I would not differentiate like this and that as two distinct clusters, but I would differentiate this and that as two distinct clusters. And it has to do, again, with the fact that you have to travel a long distance to get between those two, but not between these two. You can, inside the HC object, there is a height variable that tells you, for example, the first merge event for these guys. If I type merge, the object or the item called merge, this tells me that the very first merge event was to put individual 2 together with individual 9. So down here, it's kind of hard to tell just by looking at it, but 2 and 9, that's the lowest point at which something was joined. And the height of that merge point is equal to 17.37377. So if you were to follow this over, that is 17.37. And that is the Euclidean distance between 2 and 9. Then you reevaluate, compute a new distance matrix. Now who's closest? And it turned out that individuals 6 and 18 were closest, and they had a distance of 19.05. So this is 19.05. Then we had to use our linkage definition, because the third merge says put individual 19, a minus sign in front of these numbers means it's an individual. No, it has not been joined with anything yet. This 1 with no minus sign says whatever you created in step 1 is what you're going to join 19 with. So 19 got bundled in with 2 and 9. And the distance between 19 and the cluster containing 2 and 9, according to complete linkage, is 22.74251. We could figure that out. I'll have to do it next time because we're out of time. But if you went and got the Euclidean distance between 2 and 19, and the Euclidean distance between 9 and 19, the biggest of those two would be 22.74. One last picture that I'll show you. Those heights, if you plot the heights, HC height, it's kind of like a scree plot. The merge height for the very last merge event was up here, close to 200. So, and that's a substantial jump from the second to last one. As we go along here, these initial merges aren't very different from each other. So I don't think that these merges are defining real clusters. I think they're just putting together clusters, because these things seem to be kind of homogeneous. Once we get out here, maybe, definitely right there, there was a big difference that this last merge event put together two things that were a lot different from each other. So I would look at a picture like that to say, here's my picture. I could say there's 20 clusters in this picture. I could say there's only one. Anywhere along there I could pick it, kind of subjectively. Based on the picture that we just made of the merge heights, I would feel most comfortable just saying there's 2 clusters. That last merge event seems to be the one that was substantial. As in, the things that we put together in the last merge event, I think, are clearly different. So I think there are 1, 2 clusters. So I would do cluster analysis. I'd make a dendrogram. I'd maybe make a picture of the merge heights to decide how many of these do I want to think as being, quote, real. I think there's 2 clusters. So now I might come back and say, all right, let's pull up that metadata again. Who is column 1, or who is individual 1 and 7 and 6 and 13 and so on? Let's just do that, and then we'll be done. So meta, let's pull out these individuals from the left cluster. So it's 1, 5, 6, 7, 11, 12, 13, 18. These are all done arrays. If I pull out the other cluster, so just everything but them, it's mostly the Stambuk fuel arrays. So again, a story that's consistent with what we've seen through box plots and now principal components, now clustering. Clustering is another exploratory technique to pick up a multivariate data set and look for stories, structure, systematic patterns. I'll show you Monday if we have time, or if you want to on your own, the clusterexample.r script. There's an example in there where I create data. There's no clustering, totally random data. Run those and see what you get. Clustering is always going to give you one of these pictures. So the reason you want to make this kind of picture is because you want to know, just because there's dots or joining points in my dendrogram doesn't mean they should be joined. I could simulate data that's completely random noise and do clustering, and you'll see a dendrogram. It looks different from the one we just made, though. This one looks like there is, quote, structure, because there's at least 2 clusters. These are significantly further away from each other than everything within them. Okay, so questions? Yes. We will talk about k-means. We will talk about k-means on Monday. There will always be subjectivity involved in picking how many clusters. So yeah, we'll talk about it. It will always be somewhat subjective. You want to guard against like over-reading clustering results. What about this? Is this like some kind of weird substructure in the data? Probably not. If you have to think hard about interpreting something from one of these pictures, you're probably over-interpreting. 2 clusters is what I would say here. And I would look at the membership of these 2 clusters and try to learn something. K-means, you could try it for k equal 2, 3, 4, 5, and look at the pictures and see which seems most reasonable to your eye. There's ways to do cross-validation where you kind of let the goodness of fit guide your choice of how many clusters. You know, how well did you fit the data? But yeah, I'll do some talking about it on Monday too. Mankova. So there are model-based ways to do clustering. This is not, there's no model here, there's no statistical model. This was just an algorithm. We could say, I think my data it consists of a mixture of some unknown number of like normally distributed populations. You could say, you could assume some kind of model. If you assume a model like normal distribution or something, then you can do model-based methods like maximum likelihood and other things that would allow you to say, my estimate of the number of clusters is, you could do inference on how many clusters there are. Mankova is a multivariate technique for separating means. You could use that. That could be your model-based method. Other than that, we'd have to talk more to know exactly what you have in mind. But Mankova, regression, those types of things are ways to have a model for your data. And if you have a model, you can use it to do the actual clustering. Okay, 10 minutes late, sorry.